Good morning. At Tevard Biosciences, we are upregulating mRNA function and protein synthesis by leveraging a core molecule of the protein synthesis machinery, tRNA, and turning them into drugs. My name is Gopi Shanker. I am the CSO, and Tevard Biosciences was founded by MIT professor Harvey Lodish, along with ILP alumnus Daniel Fisher, to address some key gaps in the current slate of genetic therapies. In the recent years, we've had a wonderful set of arsenal, including gene replacement therapy, gene editing, and oligonucleotides to address many genetic disorders. And yet, there are significant gaps in these technologies and what they can do. For example, genes that are larger than 4.5 kb are very difficult to fit into AAV vectors and used for gene replacement therapy. Or genes that have complex biology and need multiple isoforms cannot be replaced. It is very difficult to control the level of gene activity when diseases are caused by so-called Goldilocks genes, where too little or too much of the gene activity results in disease, and it's difficult to control this with gene therapy or with oligonucleotides. And when mutations span the entire spectrum of a gene, it is very difficult to make gene editing practical, feasible to address the unmet need. At Tevard, we've developed a set of 3 platforms that can really address this problem by leveraging tRNAs. Our suppressor tRNA platform focuses on diseases caused by nonsense mutations and can really allow the normal functional protein to be formed from the mutated copy of the gene. And our enhancer and amplifier platforms target diseases that are caused by haploinsufficiency, where one copy of the gene is mutated, but the other copy is functional, and we can upregulate protein synthesis from the functional allele to compensate for the lost protein. Today, let me give you a little bit of a vignette into one of those platforms, but let me tell you some unique attributes of our tRNA-based therapies. Like gene therapies and oligos, we can really address the root cause of the disease, but we can be agnostic of the size of the target gene because tRNA genes are very small and can be packaged very efficiently into a wide variety of delivery vehicles, so the gene size doesn't matter. One of our platforms that I'll tell you about, suppressor tRNAs, can really be expanded into multiple indications where a single drug can be used for multiple diseases, which is a really unique concept in the space of gene therapies. Through viral vectors, we can use a single-time delivery, but most importantly, because we leverage the normal expression patterns of the mRNA by the cells, we can restore the mRNA expression and protein synthesis to normal levels in cells that normally express these genes, thereby maintaining the kinetics of gene expression and balancing efficacy with safety. So let me tell you a little bit about our suppressor tRNA platform. Somewhere from 1/4 to 1/3 of all disease-causing mutations occur when one of the amino acid coding codons gets converted to so-called premature stop codon. As you all know, normally, these stop codons occur at the end of protein coding sequences and code for translation termination and end of protein synthesis. But when a mutation results in a stop codon in the middle of a gene, that results in truncated non-functional protein. What we've done at Tevard is we've engineered tRNA molecules that normally code for amino acids to recognize such a premature nonsense mutation, incorporate the correct amino acid, and thereby restore normal functional protein from the mutant allele. One of the really unique attributes of this technology is that when one looks at the frequency of nonsense mutations across the entire spectrum of disease-causing genes and disease-causing mutations, only a handful of these mutations occur very commonly. For example, arginine codons converted to TGA or glutamine codons converted to a UAG stop codon account for almost half of all disease-causing nonsense mutations. And these suppressor tRNAs can be highly mutation-specific, but can be gene context agnostic. What you see on the right half of the slide is a dozen genes where mutations lead to a variety of epileptic syndromes. And we can develop suppressor tRNAs that target one or other commonly occurring mutation and can treat subsets of patients with these mutations across all of those epileptic disorders, which means a single tRNA drug can treat mutations in virtually any gene that has that mutation. Doesn't matter what the indication is and doesn't matter what the gene itself is. So true small molecule and antibody-like indication expansion for a gene therapy. pretty unique. So what we are here today is to explore opportunities to leverage all three of our unique platforms, and I only got to tell you about one, so please come and meet us at the booth next door so we can tell you about the rest of our platforms as well. We have a foundational partnership with UCB Pharma to advance epilepsies, but we've demonstrated proof of biology and mechanism in animal models across multiple indications. We have programs in CNS, cardiology, and musculoskeletal disorders, but we're interested in additional areas because our platforms are disease area agnostic and target gene agnostic. And we'd love to explore partnerships that are either focused on disease areas so that we can advance these novel therapies for those patients who desperately need them, or to partner to access next generation viral and non-viral delivery vehicles so that we can get these really unique cargoes to the patients that need them so desperately. We are right across the street on Main Street from this hotel, and please come and meet us, and so that we can talk about how to take these to the patients. Thank you.